Dear students, in this episode, we'll discuss other phytopharmaceuticals of industrial importance. It has been divided into 5 modules. Module 1, Mentha and Menthol, in which success story of India in producing menthol has been discussed. Module 2 covers extraction and isolation of opium alkaloids and morphine. Module 3 extraction and isolation of alkaloid quinine from cinchona bark. Module 4 extraction and isolation of reserpine from Rauwolfia. And Module 5 a brief account of isolation of sorrelin, a furanocoumarin. Essential aromatic or volatile oils are generally used as flavoring agent in pharmaceutical preparations. India produces and exports quite a good number of essential oils which are used as flavoring purposes in pharmaceutical industry, food industry and cosmetic industry. Council of Scientific Industrial Research laboratories and other national laboratories are doing commendable work in this field. Module 1, Mentha and Menthol. India exports different types of mint oils to a number of countries including Argentina, Brazil, France, Germany, Japan, UK, USA, etc. These varieties include the Japanese mint oil derived from Mentha arvensis, peppermint oil Mentha piperita, dementholized Japanese mint oil, spearmint oil Mentha spicata, water mint oil Mentha aquatica, horse mint oil Mentha sylvestris, bergamotol oil Mentha citata, and still others. India cultivates about 8 species of mentha. However, 3 species are approved or recognized for the purpose of quality standards and international marketing. Japanese mint oil is not distinguished from the peppermint oil in the Indian trade. Menthol production in India. The cultivation of Mentha arvensis in India is a dramatic and very successful story. Before 1964, there was no production of mentha oil and menthol in India. The cultivation of Mentha arvensis was started around 1870 in Japan, so it is called Japanese mint, then extant in Brazil, China and Formosa, etc. Colonel R.N. Chopra and Dr. I.N. Chopra of Regional Research Laboratory Jammu Tawi first brought Mentha arvensis in 1964, and with the best efforts of Mr. S.C. Bante, Managing Director, Hindustan Richardson Limited, the commercial cultivation could be started in India. The survey conducted by Richardson Hindustan Limited determined that Tarai area of Uttar Pradesh would be a suitable place for the cultivation of Mentha arvensis. So, a land of 15 hectares was purchased to set up an agricultural research center for Mentha. The crop of Mentha was successfully grown. When the Mentha crop became available, Hindustan Richardson Limited established a distillation unit in Bilaspur of district Nainital to extract Mentha oil by steam distillation. This built confidence in the farmers to grow Mentha herb. They also supplied the planting material as well as giving technical knowledge to grow the Mentha herb. As a result, the area of Mentha crop subsequently increased in entire Tarai and western districts of UP, mainly Nainital, Rampur, Moradabad, Badaयूं, Bareilly, and recently extended to eastern districts, for example, Barabanki, Lucknow, Sitapur district, crop of India grown in Uttar Pradesh. Rest 20% in Punjab, Haryana. Crop of Mentha herb were also attempted in MP and Gujarat, but in little quantity. With the best efforts of farmers, small-scale industries, and CSIR laboratories, RL Jammu, CMAP, Lucknow, India is one of the largest producers of menthol and mint-allied products in the world. Now, second one is China. Presently, 85 productions of mint products in India rest in China, Brazil, and other small countries. India dominates global production with around 80% of global supply, followed by China and Japan, each producing around 10%. India exports around 25 to 30,000 tons in a range of forms: menthol crystals and powder, demantholized mint oil, arvensis oil, etc., with the balance of production used domestically. India's domestic consumption accounts for around 40% of global consumption, with China 20%, Europe 1.5%, with Germany and Netherlands the major users, and the USA 15% according for the bulk of consumption. Distillation and processing of mentha oil in India. Distillation and processing of mentha. The oil derived from the mentha arvensis herb, mentha oil, by steam distillation, which is the basic raw material required for the mentha manufacture. By freezing, that is chilling, of mentha oil, menthol crystal or flakes are isolated in India. There are two types of products: menthol crystal and flakes. Some manufacturers are processing to their shape and size: bold, that is large crystals; medium crystals, small crystals; flakes in shape of powder; rest mother liquored by centrifuging cause demantholized peppermint oil. Menthol is main constituent in the mentha oil. Rest contains menthone, methyl acetate, neomenthol, mint terpenes, alpha-pinene, beta-pinene, L-limonene, 3-octanol, piperotin are also main contents of the use as raw material to produce value-added products for flavoring and fragrances industry. Menthol mint essential oil is the main source of natural menthol. The demantholized oil is reconstituted to produce oil similar to mentha piperata, Several of the many dozens of terpenes present in the essential oil are valuable compounds that find uses in cosmetic and pharmaceutical industries. Menthol itself is used as a flavoring agent, mouth-related produces such as fresheners, toothpaste and chewing gums, etc. Menthol, mint oil, because of the antimicrobial properties, enhances the shelf of edible products and grains. Menthol mint oil is an essential ingredient of the ointments, pain balms, lozenges and syrups. It is used in cosmetic preparations, cologne, deodorants, cosmetics, etc. in India. Menthol and mint oil are extensively used in betel, that is palm, related industries. Menthol is generally obtained by chilling mint oil and subsequent centrifuging to separate out the menthol crystals. The process may be divided into two steps. Firstly, menthol flakes are produced by chilling mint oil, and secondly, menthol flakes mixed with mint oil again chilled to produce bold crystals of menthol. The total recovery of menthol in the first step is around 70%. The time cycle for first step is around 67 days. In the second step, menthol flakes mixed with certain preparations of mint oil kept under temperature control for about 25 days. The mother liquor, that is concentrated mint oil, is a type product and can be used again till discolorization of oil occurs. Total recovery of bold menthol crystal is about 50%. The details of cost involved in the distillation of menthol oil by farmers were also analyzed, and it was revealed that small units of menthol distillation, large farmers did not go for higher distillation as all of them had their own distillation unit. The economic aspect of menthol production is utilization of byproducts, which may be summarized in the following chart. Mentha arvensis herb is steam distilled, and thus we get mentha oil and spent hay. Spent hay is further utilized as a fuel for heating the still. The mentha oil is then separated in crude L-menthol and residual oil when it is chilled. The residual oil after chemical treatment gives us menthol, which is a byproduct 1, and menthol oil on chilling gives you crude L-menthol, which is again supplemented by the crude L-menthol obtained from residual oil. From this crude L-menthol, pure L-menthol bold crystals are obtained, and menthol isomers are the second byproduct of this process. India exports different types of mint oil to a number of countries including Argentina, Brazil, France, Germany, Japan, UK, USA, etc. These varieties include the Japanese mint oil derived from Mentha arvensis, peppermint oil, Mentha peperita, dementholized Japanese mint oil, spearmint oil from Mentha spicata, water mint oil, Mentha aquatica, horse mint oil, Mentha sylvestris, bergamot oil, Mentha citrata, and still others. India cultivation about 8 species of mentha. However, these species are approved or recognized by the purpose of quality standards and international marketing. The nature of oil yields on an average 40 to 50% menthol and 50 to 60% of dementholized oil, which can be used both in confectionery and medicine in place of imported peppermint oil. Japanese mint oil is not distinguished from the peppermint oil in the Indian trade. The analysis and production data of industrial oil are shown in the following charts. Source cultivation and utilization of aromatic plants RRL Jammu. Module 2. Isolation of morphine. Morphine is the principal alkaloid which occurs in opium to the extent of 8 to 15%. Opium is the dried exudate which is obtained when incisions are made on unripe capsules of Papaver somniferum. The cultivation, collection and processing of opium is under international controls as opium and products obtained by its processing are prohibited because of the health hazards associated and their use for recreational and addictive uses. Legal opium production is allowed under the United Nations Single Convention on Narcotic Drug and other international drug treaties, subject to strict supervision by the law enforcement agencies of individual countries. Opium is cultivated under government licenses at designated places and its processing, sale, transport and all other related operations are offences if not covered under suitable licenses. Although opium is an illicit product, its alkaloids, for example morphine, codeine, noscapine and others, are widely utilized for therapeutic purposes. Morphine being an analgesic, the use of which cannot be dispensed in certain clinical conditions. Opium, poppy, is a dried latex obtained from the opium poppy, scientific name Papaver somniferum. Opium latex contains approximately 12% of the analgesic alkaloid morphine, which is processed chemically to produce heroin and other synthetic opioids for medicinal uses and for illegal drug trade. The latex also contains the closely related opiates, codeine and thebaine, and non-analgesic alkaloids such as papaverine and noscapine. The traditional labor-intensive method of obtaining the latex is to make incisions on the immature capsules, fruits, by hand. The latex leaks out and dries to a sticky yellowish residue that is later scraped off and dehydrated. The story of the isolation of opium alkaloid is very old, but one can start with late 18th century and early 19th century when William Gregory started taking interest in the chemistry of opium alkaloids. Schertner, the pharmacist, isolated morphine in 1805 and supplies were associated with another analgesic but less potent alkaloid, codeine. Gregory in 1831 published a process for isolating morphine hydrochloride in high state of purity. The process consisted of concentrating an aqueous extract and precipitating the alkaloids with ammonia. The alkaloids were collected and neutralized with hydrochloric acid, and after staining, a mixture of hydrochlorides crystallized out. This became known as Gregory salt and contained codeine, which was later isolated by Robiquit. Modified Gregory processes are employed by different manufacturers of opium alkaloid. Changes have been made from time to time, and two types of processes are outlined in charts 1 and 2. In the first process, boil opium in water and add calcium chloride and then filter. It is then divided into mark and filtrate. Mark contains narcotine and meconate, and filtrate is evaporated in vacuum to thick magma. On cooling, it crystallizes. Separate the thick mother liquor under pressure, and again you will get two fractions, the mother liquor fraction and crude Gregory salt fraction, which is washed. It is then dissolved in hot water and treated with alkali. On staining, the morphine alkaloid precipitate, they are then converted to salt and decrystallized salt till they are pure. The mother liquor solvent extraction is undertaken, and in this mother liquor, codeine is there. It is recrystallized till it becomes pure, and other alkaloids also, after recrystallization, give codeine. Thus, in the two fractions, we get morphine and codeine in the crystalline forms. In the second process, opium is boiled in water and filtered, and thus we get again two fractions as above. The mark contains narcotine etc., and the filtrate contain other alkaloids which are evaporated in vacuum and cooled. This is treated with alkali, and we get crude alkaloid precipitate, codeine in liquor. The crude alkaloid precipitate is extracted with solvent, and then we get crude morphine on evaporation of solvent. In solution, you have morphine. This is converted to salt and recrystallized till it is pure. In the liquor, you have codeine and other alkaloids in solvent. Solvent extraction and press treatment, we get codeine, which is recrystallized till pure, and other alkaloids also, which are subjected to different treatment to get codeine. The processes are essentially similar. The processes outlined were employed by various manufacturers during past years, including government opium alkaloid factories, are now obsolete and are being replaced by newer technologies which are trade secrets. The modern processes may be summarized as follows. Opium is digested with dilute acids like acetic acid or sulfurous acid. The mark is rejected and the alkaloid separated by solvent extraction at different pH based on their substitution pattern, oxidation states, and dissociation constituents pKb values. Both cation and anion exchange chromatography have been successfully applied to the isolation of morphine from opium. Morphine, the free base, unlike other alkaloids in free base forms, is only sparingly soluble in chloroform and nearly insoluble in ether or benzene. Similarly, solubility of morphine in sodium hydroxide or potassium hydroxide solutions differ considerably with that of codeine and thus utilized for their separation. The following schemes outlines separation of different alkaloids of opium. Powdered opium is taken and it is shaken with calcium chloride solution. After that, it is filtered. We get insoluble matter which is discarded and the filtrate which contain hydrochlorides of alkaloids. The volume is reduced by evaporation under reduced pressure to a syrupy liquid, and then 10% sodium hydroxide solution is added. Addition of sodium hydroxide solution causes precipitation of alkaloid, and this precipitate contains of nascapen, papaverine and thevenin, and in alkaline solution are morphine, codeine and narcine. The precipitate is dissolved in dilute alcohol and then acetic acid is added to make it slightly acidic. After that, about 3 volumes of boiling water are added. On addition of water, a precipitate is formed which contains nascapen and papaverine, and in solution remains thevenin. The precipitate is dissolved in boiling 33% oxalic acid solution and allowed to stain. The solution is brought to boiling, again allowed to stain, again brought to boiling, and this process is repeated 2 to 3 times. Finally, when it is allowed to stain, we get crystals which are of papaverine acid oxalate and solution which contain nascapic acid oxalate. A solution is made alkaline with ammonia, a precipitate is formed and the solution is discarded. Nascapen is dissolved in boiling alcohol and after crystallization we get crystals of nascapen. The alkaline solution which contain morphine, codeine and narcine is extracted with chloroform and we get chloroform extract and aqueous alkaline solution. The chloroform extract contain codeine whereas aqueous alkaline solution contain morphine and narcine. The aqueous alkaline solution containing morphine and narcine is made acidic and we get aqueous acidic solutions which form salts of morphine and narcine. This is made slightly alkaline with ammonia, and when ammonia is added we get precipitate which is morphine, and this is subjected to further purification. The solution contains narcine which again is further purified. The chloroform active contains codeine and from this codeine is subjected to further purification.